recording because Riso said
okay.
that we have to record and send for his.
Did
Okay,
you?
sorry,
Huh?
Maria Clara. Yes,
Okay,
I
sorry.
did.
So the International PhD School on Advanced Oxidation Processes is an initiative of academics and scientists with a strong and internationally recognized expertise in the environmental applications of AOPs. The aim of the school is to bring members of the scientific community themselves on a voluntary basis in promoting higher education of young scientists in this research field. The school was founded in 2014, but then in 20 only for European students, but then in 2018 it became international with the inclusion of students and professors from Latin America. It promotes very many different events such as summer schools and the series of webinars, opportunities for networking for school members through calls for joint proposals, mobility among PhD students between different universities which are member of the international PhD schools. And also it enables a label, which is the International PhD on AOPs. So even if you did your PhD only in Latin America, for example, you go through an evaluation process and you can have a label as an international PhD on AOPs for your thesis, which is really nice afterwards to get international recognition and also the best PhD thesis awards. So on behalf of the IPS AOP school, today I'm very pleased to introduce you to our speaker, who I admire a lot. So Professor Camila Costa de Amorim is a professor at the Federal University of Minas Gerais. She is an environmental engineer who studied at the Federal University of Viçosa. She has a master's and a PhD in sanitation, environment, and water resources. She is a professor at the Department of Sanitary and Environmental Engineering at the Federal University of Minas Gerais since 2010. And she's involved with teaching and research activities. She's the leader of the research group on environmental applications of advanced oxidation processes. And she guides research on the development and application of advanced oxidation processes applied mainly to water and wastewater treatment in either domestic and industrial and also hospital effluents at the moment, with treatments focusing on disinfection, removal of antimicrobial resistant bacteria and genes, and also contaminants of emerging concern to improve wastewater quality for reuse in agriculture and also discharge. So Camila, the floor is yours. You're welcome to give your presentation.
Okay, thank you, Maria Clara. Don't.
I just would like to ask everyone to keep the microphone off during your
Okay.
presentation.
Thank you to introduce me. I wanted to say for everyone that it's a pleasure for me to be here. It's like really interesting to participate in of this this plenary because this is an initiative of AOP schools, and Riso is doing a very good job with this AOP schools and with this webinar. But the challenge here for me today, it's big, it's very big, and then I will try to do my best, but to be honest, it's very complicated to stay here with the monsters of the research, like Despo and Pulgarin and Riso as well. So they are like my inspirations and the inspirations of many students, PhD students around the world. And for my research group as well, they are the inspirations. And thanks a lot to stay here today with us. And don't expect very good English like Maria Clara, okay? It's not fair with me, but I will try. So I think that we can start. Um, I will share my presentation. Just 1 second. Is here. Okay. Can you see?
Yes, we see your presentation.
Yes, the presentation or the. I think that I have to change just the screen. 1 second, please. I will share the another one. This one. Yes, just confirm. Can you see the presentation now?
Yes. I don't know if you are in the first slide though.
Yeah, I'm not. Second. Yes. Now. Okay. It's
That's it.
working now?
Yes, perfect.
Okay, fine. So, um, as Maria Clara said, I am a professor in a Universidade Federal de Minas Gerais in Belo Horizonte, Brazil. And Luigi give me like a big challenge to present this theme, this about the effect of AOPs on antibiotic resistance. And this is the part of the webinars, as Maria Clara introduced. So, for this presentation, I will...
Fíjate, ¿tú sabes cómo se hace una captura de pantalla?
Sorry?
Vale, sí, esa la conozco estupendo.
Sorry, Luigi.
Sorry, I muted him already.
Yeah, okay. Thank you. So for this presentation, I will try to have a brief introduction about the problems that will be present here, and I will talk about the antibiotic resistance and the most important research that um, are published in this area, and about to AUP to combat and prevent the antimicrobial resistance. Finally, I will present our current results about the treatments of wastewater treatment to combating antimicrobial resistance in Brazil. So the problems related to clean water and access and waste still remain even in 21st century. We know that we have a lot of problems about the water quality to waste disposal, and this is more emphasizes in our country, in developing country. And now we have new concern about the presence of micropollutants or contaminant of emerging concern. And the antimicrobial can be found in different environments, adding to antibiotic resistance, and then the antibiotic resistance now it's like the new challenge for, and is considering as a contaminant of emerging concern as well. So the problems is because nowadays they are not only more worried about the presence of these antimicrobials only in the clinical or medical um, areas, and not only the human habitats, but in other habitats as aquatic habitats and terrestrial habitats and building habitats as well. So the antimicrobials play a critical role in the treatment of human and animal disease. It's well known for everyone. And the prescriptions of this kind of chemicals are very high around the world. And this is the big problem because when we take it in the antimicrobials, we can spread this, we can discharge this with that some parts are not or consumption with our organism. So The antimicrobial, they estimated that in USA over 250 million of antibiotics prescriptions annually, and around the world 70 billion prescriptions per year. In 2017, the World Health Organization published a list of 12 bacteria families that are considered resistant to the most powerful antibiotics. So globally, the antimicrobial resistance leads to, they estimated that they leads to a 700,000 deaths per year. So the antimicrobial resistance, this is appear like because the use of antibiotics, as I said, and this can also cause antibiotic resistance bacteria and to further spread antibiotic resistance genes. So antibiotic resistance genes have also emerged as an environmental contaminant, and these genes have the capacity to spread among the bacteria and distribute from human and animal source to natural environments and drinking water resource as well. The last publication from this year in the Lancet, in the chapter of antimicrobial resistance collaborators, they estimated in this report the global research on antimicrobial resistance that estimated that 1.27 million deaths in 2019. These deaths were directly attributable to a drug-resistant infections, and 4.95 million deaths that were associated with antimicrobial resistance. So in 2017, and I said the World Health Organization published the prior pathogen list like that needs like urgently action about the mainly pathogens that it's spread and there is in the environment to and this exercise is necessary to combat the dissemination of these pathogens. So recent publication in Nature Communication, they investigated with with a lot of data around the world, the presence of these genes, the antibiotic resistance genes in different habitats like air, aquatic, human, terrestrial, and others. And then they proved that we have different antimicrobial resistance gene classes, not only in human, but even in aquatic habitats. And this is important information for us because we are investigating the removal of these bacteria and resistance bacteria and genes in aquatic habitats, and the beta-lactams classes, antibiotic classes, is like the most found in these habitats, in this environment. So what about the antimicrobial resistance in wastewater treatment plants? These are considered the hotspots of the for release the contaminant of this kind of contaminants for the environment, because the presence of the antibiotics is present in subinhibitory concentrations, and then we have the biological reactors in the most part of these plants, the wastewater treatment plants, municipal wastewater treatment plants. So we have the abiotic factors and biotic factors that could allow the development or the increase of the genes transfer and the spread of antibiotic resistance bacteria and genes. And of course, we have a plenty of publications, very important publications. And over here with us, we have the Expo Facta Casinos, and we have publications, very important publications with Luigi Rizzo as well, and Bulgarin as well, and other authors. And they have been studying the occurrence of this kind of bacteria and genes in different environment, and even in the wastewater treatment plants. But what about the conventional treatment steps, the conventional treatment strategies? So some studies, some like more old studies that identify that the inactivation of the common indicator bacteria like E. coli may not guarantee the activation of the intracellular antimicrobial resistance genes. So even after bacteria are greatly inactivated, they prove that DNA activity may remain. In this direction, we have to investigate the best option to treat this kind of wastewater to guarantee that we are not increasing the gene transference or the mutation, or if you are deactivating the genes and not only remove the bacteria. So the strategies for treatment could have to involve the characteristics of the oxidant, and the oxidant, despite of the advanced oxidative process, they are very reactive species with the formation of hydroxyl radicals. But the oxidant have to move through the cell membrane to achieve the DNA and not undergo extensive binding to other cellular constitutions. Um, we know that we have cell membrane with gram-negative and gram-positive, and Gianax Bulgarin have a very interesting publication with this matter. And the oxidant have to be present in sufficient quantity inside the cell to react with DNA that contains ARGs. So in this direction, we have kinds of oxidants that could only achieve the A area that is in this figure, or the oxidants that could um, direct achieve the cytoplasms, including DNA. So we have difference with gram-positive cells that have a thick peptidoglycan layer, while gram-negative cells that have a thin peptidoglycan layer. So the studies about how this could guarantee not only the disinfection or the removal of bacteria, but even the deactivation of the DNA. And a recent study in 2018 from DESPO and collaboration and colleagues, they have a very big review and a very good review about the kind of inactivations of ARB and the removal of ANGs by advanced oxidative process. And then they conclude that the inactivation of this depends, of course, of microbiological factors and waste and like treatment, the factors that involve the treatments, the operation of the treatments. So they emphasize that we have to take care about the operation conditions like disinfection dose, time, contact time, and the wastewater composition, then the matrix composition. And then we have to pay attention about the specific oxidative damage mechanisms about the treatment technology. Or like which which kind of technology could be used in according the characteristics of the wastewater. And then we have to consider the type of bacterial species and genes under study and antibiotics as well, to which resistance is acquired. So I would recommend that everyone, like every PhD students that are studying this subject, to see this publication, because it's there is a very good result here. And a very fresh publication from HISU, like a critical
Camila,
review.
you're not
Yes?
sharing your PowerPoint anymore.
No?
No.
I'm not sure what has happened. And now,
Yes.
yes, thank
Mhm.
you. So a recent critical review from HISU. It's like how we can use the AOPs as tertiary treatment. So we know that conventional wastewater technology are ineffective to remove and inactivating ARB and ARGs. So the advanced oxidative process could be a good choice to use in this matter. And he's pointed very specific not problems, but the advanced and drawbacks for the use of each kind of AOPs. If you are going to this critical review published by HISO this year, we have a table that he put it different kind of advanced treatment, and then they put it photophantone as well. And we here in Brazil, we already worked with a lot of advanced oxidative treatments, but nowadays, in like maybe 3-4 years later, we start to investigate the photophantone. Why we started to investigate this kind of process? Because in our country, we have a lot of problems with the technological, not capacity, but resource and infrastructure, and we have a plenty of sunlight all the year. So we believe that this process could be applied in our country, and maybe the some drawbacks that Risa pointed here about the large surface areas. Yes, it's true, but in Brazil we have some large surface areas yet, and I know that could be a really drawbacks, and I would like to pay to give attention and the drawback to operate the process at neutral pH, like the additional of chelating agents to possibility uh, to operate in the pH near to the wastewater effluents, the wastewater secondary effluents from municipal wastewater treatment plants. In our research group, we are adopting the strategies to avoid the use of these chemicals agents, because we agree with this review that this could increase like the carbon and then the cost of the operation, of course. So we are adopting the strategy to intermittent iron additions, but we are always worried about the quantity of the iron that we have at the end of the process. But I will talk more about that in some minutes. So for about the AOP technology, we have a plenty of publications and a plenty of manuscripts about that. And for photophantom and phantom reactions. In the past, like in the beginning of when they begin to study that, they attribute, like they consider the non-selective and very reactive from hydroxyl radicals formed in this reaction. When you have iron and hydrogen peroxide, we can generate this radical oxidant species that could deactivate ARG because this oxidative species can directly attack the open circular DNA, leading to DNA fragments. But recent published for Janax and Bulgarin, a very interesting manuscript, they identify like the both mechanisms and intracellular and extracellular pathways to deactivated bacteria and to removal bacteria from photophantom treatment. So Gianax and colleagues and Bulgarin as well, they studied about the photophantom process and they found to be essentially an intracellular process inside of the bacteria. And then this external radical oxidative species actions, um, it's contribute of the process as well, but the main, how can I say, I could say important will be about the intracellular pathways. Uh, Continues with the effect of photophantom reactions on antimicrobial resistance. We have other publication about the effects of the solar photophantom, and again we have a Bulgarin and GNX. When they tested the both solar lights and solar photophantom process to eliminate an ARB, and they have tested with different bacteria in water. In wastewater, they conclude that the both processes were effective to reduce antibiotic resistance genes, although solar photophantom was more rapid than solar light. The solar photophantom process ensure effective disinfection of ARB and elimination of ARGs, and it's a potential means to ensure limitation of ARB and ARG is spread in the nature. In nature. So, according to the results, we start to investigate in the application of these photophantom reactors with the secondary or with the secondary effluents from Brazil. In our country, we have a very big challenge with the wastewater treatment because we have a low coverage of the treatment. So only less than 62% of the population has the sewage collected, and we have a very big country. Most of the parts of the country, we have just the disposal in the soil or in the, yes, direction the soil and some specific compartments, and less than 43% of the population has the treatment. Most wastewater treatment plants operated until the secondary stage of the treatment, the tertiary treatment. It's far from our reality. And many wastewater treatment plants operated under inadequate conditions due to financial, technical, and low maintenance limitation. And even when we have well-operated wastewater treatment plants, the removal of these emergent pollutants is not satisfactory, leading to phase transfers being absorbed in the sludge or maybe is disposed with the secondary effluent. So, because of that, we believe that to use solar technologies could be a good point, could be a good option to better, how can I say, to increase or to develop, to better develop the wastewater treatment in our country as a tertiary treatment, like to not only contribute to remove the emergent pollutants, but also to contribute to have better effluent quality for disposal or agricultural reuse. Here we have, I will present like some results from our research group. These results, it's from what is the diversity of the bacterial community in the wastewater in Brazil. This is all of this data is from the effluents, real secondary effluents collected in Brazil, in Belo Horizonte. This wastewater treatment plants treats the wastewater from 1.5 million of inhabitants. It's a considerable, it's a very like considerable wastewater treatment plants. And we have collected the samples from dry season and wet season. We had collected 11 samples, and we did the sequencing of these samples, identifying the bacteria community. We can see here we didn't have the difference in dry and wet season about the bacterial diversity. And then we can see the predominance of proteobacteria, as actinobacteria, and bacteroidetes. We have almost 30% of proteobacteria and 20% and of bacteroidetes and actinobacteria. These results, it's corroborating with the another results from another parts of the world that are investigating the community, the bacterial community in the wastewater. This is a secondary wastewater. This wastewater was pre-treated by the conventional activated sludge system. So when we investigated the associations between the ARG subtypes and microbial community, we aimed to investigate the correlation between the World Health Organization priority pathogens detected in our samples of the wastewater treatment plants in Belo Horizonte. So we could identify the most present, the priority and the critical priority and high priority that the World Health Organization published, the list of the World Health Organization. So we can identify the multi-drug resistance for Actinobacter, Baumannii, Pseudomonas aeruginosus, and the Enterococcus as well. And we can see this in the Enterococcus vancomycin resistance and Actinobacter baumannii to carbapenems resistance that could be identified in our samples. And this where was done, this was achieved after the WGS sequence of our samples. So the first of all, we aimed to identify if our treatment could not only remove bacteria, but also prevent or inactivate the plasmids that could conferring like transfer genes transfer to non-resistant cells, like when you have the migration of the plasmids and this cell replication could create or generate other cells with the resistance genes. So this work we taken two plasmid DNA. One is resistance to kanamycin. And then we put these plasmids in the secondary wastewater and applied the solar photophantom, and we investigated the transformation, and we investigated with the if the plasmid was inactivated and if the plasmid could transfer the genetic material for another bacteria. So we put it together and non-resistance bacteria, and E. coli, and we investigated out and also investigated if we could have the transfer of this genetic material. So the results shows that we have the complete plasmids inactivation after 6 minutes of solar photophantom. We can see here our results. And we had all sets of control experiments as well. So we used 50 milligrams of hydrogen peroxide with the strategy to intermittent iron addition. So we can see the results of the consumption of hydrogen peroxide and iron as well. And then we could prove that our treatment was not only effective to inactivate the plasmid, but to have the complete inactivated of the resistance E. coli. So we put these plasmids with the E. coli non-resistance. So this is a positive control and negative control. And after the treatment, we could identify this removal, the totally removal of the plasmids with 60 minutes. So for hydrogen peroxide and solar, the process were also efficient to inactivate the plasmids in E. coli. So these results corroborating with the results from Gianax and Pulgarin that trying to explain like the efficiency of just sun and photophantom, solar photophantom as well. With another results, we have we investigated the effect of solar photophantom on antibiotic resistance, and we did the metagenomic analysis of our wastewater, secondary wastewater, and we tracked the samples now were treated by solar photophantom at neutral pH without complex agents. Okay, we just use the intermittent iron addition. So here we have again the effect of the solar photophantom on bacteria phyla and bacteria communities. And we can see here only the samples from the secondary effluents, secondary wastewater, and the different types of treatments with the controls as well. So for solar photophantom, we use 30 milligrams of iron and 50 milligrams of hydrogen peroxide, and we achieve 6% to remove of acidobacteria phyla, 8% for chloroflexi, almost 8% for actinobacteria, and 76% of bacteroidetes and 74% for Firmicodes. But on other hand, we have enrichment of proteobacteria in 4 times. According to the results for genes removed, we achieve more than 6% of removal of ARGs for genes for sulfamet genes for here macrolides, tetracyclines, and beta-lactams and fluoroquinolones as well. I have highlighted these areas that we could emphasize the enrichment of the proteobacteria with this treatment, and even in the presence of hydrogen peroxide. So these results, we believe that the proteobacteria selections was also observed in other publications, as in after ozonization of municipal wastewater, and suggests high resistance of this group to oxidative conditions. Moreover, we identify we just investigated the proteobacteria classes, and in this graphic, we can see only the proteobacteria classes from the bacteria community, and we identify 5 major classes and in the Proteobacteria phylum. And we identify the high increases in solar photoplankton samples, and uh, when you have the presence of hydrogen peroxide, it was observed for gamma proteobacteria, mainly with the genera Pseudomonas and Streptomonas. So these genera are also important because they are the Pseudomonas aeruginosus are a critical species, like from the WHO priority lists. And just my mouse is yes, and we know that we identified relative abundance of this gamma proteobacteria class also increased in the presence of only hydrogen peroxide and after solar-driven AOPs. These results corroborating again with other authors that investigated the photoplankton reactions in the presence of the bacteria. So when we are going to identify this, the what is the reasons for this enrichment, we can see that some kind of genes present with the stenotrophomonas, they are they have hydrogen peroxide scavenging mechanisms, and the genes that I put it here in the presentation were present in our samples. So for that genes, specific genes, it's well known that they played a critical role for survival in the presence of high hydrogen concentrations, and our samples, solar photoplankton and control phantom samples, present 4 or 5-fold growth in these kind of genes. So we can see here that we have the same increase of genes that are well known to have the scavenging mechanisms, and this could interfere in these results with our treatments. So suggesting a significant adaptive response of hydrogen peroxide. On the other hand, we're trying to investigate alternative oxidants. So we know that persulfate now is an interesting alternative oxidant because they could stay more time in the environment, and they could promote less strong reactions, and they could maybe have more contact time with the bacteria and genes. So in our results for investigating the bacterial community, we can see the same bacterial phyla, but we have different efficiency removal for bacteroidetes, and now we have the enrichment of proteobacteria and Firmicutes when you use the both oxidants, hydrogen peroxide and persulfate. So when you use the only persulfate in solar photoplankton process, we have 88% to remove of proteobacteria, but we have an enrichment of Firmicutes that we can see here. So the impact of this persulfated in photoplankton in solar photoplankton proteobacteria, it was promised, and it was good because another oxidant, we had the enrichment, but on another hand, we have the enrichment of the Fermicodes. And when we investigated the enrichment of the Fermicodes, uh, we we achieved that the main causes of this enrichment could be because the cell membrane. So Firmicutes are mainly represented by gram-positive bacteria which have a thicker and more dense membrane layer compared to gram-negative bacteria. So we believe that the exposes of these bacteria for these extreme conditions or for these conditions, the difference with the membrane layer could have more capacity of this bacteria to to be exposed to extreme conditions. So we always also investigated the elimination of the whole priority pathogens removal with this treatment, a photophantom, solar photophantom, and we identified the removal of 5 World Health Organization priority pathogens, including Enterococcus, Staphylococcus, and Klebsiella, and the critical priority of Pseudomonas aeruginosa and Staphylomonas maltophilia, and emergent pathogens responsible for morbidity and mortality were both totally removed by our process, our treatments, solar photophantom treatments. Yet, according to the investigation of effect in ARB deactivation, we identify that solar photophantom eliminated 100% of ARGs that with the fluoroquinolones and antibiotic and beta-lactams antibiotic resistance. Like here, we have the classes of found in the secondary wastewater, and here after the treatment with phantom and solar photophantom. These were investigated with WGS sequence, and we found a greater ARGs diversity detecting our samples without treatment, and we detected genes that confer resistance to sulfonamides, macrolides, tetracyclines, and amyloglycosides, corroboration with another results from other countries. And the solar photophantom removal nearly 60% of ARG associated to sulfonamides, macrolides, and tetracyclines, with completed removal of beta-lactams and fluoroquinolones. When you use the alternative oxidants, we have the difference between the treatment, because when you use the combination of solar photophantom and persulfate, solar with persulfate and hydrogen peroxide, we could we didn't could identify any genes and resistance genes after the sequencing. So we didn't have more genetic material to sequence. So this means that our treatment was effective to remove the genes when you combine the both oxidants. We have here some elimination of macrolides and bacitracin again, and tetracycline again. And finally, when we are investigating the treatment with the solar photophantom using only persulfate, it's not the combining the oxidant, but only the persulfate, we could have very good results as well. Like we could eliminate more than 50% of some genes, and in the median we could eliminate 45% of the all of the genes, resistance genes that were present in our sample. Finally, nowadays we are working on the full scale of these technologies, and we already in like a few weeks, I hope, we will publish a new paper with the practical and real application of these treatments. And then we are working on a scale of this technology. So in Brazil, we constructed our CPC reactor and RPA reactor, and now we are trying to put it inside of the wastewater treatment plants to have more realistic results. So very soon we will have these results published. I will just put it here like to emphasize. And as conclusions of our work, we can see that solar AOP applied to real secondary wastewater to analyze treatment effect of antimicrobial resistance bacteria and genes are effective. Solar photophantom reach more 99% of plasmids that carry antimicrobial resistance and which may contribute to the spread of ARGs. Solar photophantom using intermittent iron additions and with 30 milligrams per liter and 50 milligrams per liter of hydrogen peroxide was the most effective treatment on the removal of AMB and ARGs. And the combining of the alternative oxidants, the combination, that could be a good option to combat the genes. This treatment completely fragmented sample DNA, hiding WGS analysis. Moreover, I would like to emphasize about the critical review from results from this year uh, that he put it the AOPs like the challenge of 10 recommendations for to promote the AOPs as tertiary treatment. And from these 10 recommendations, I was reading and I was looking that we were working on the 6 of these 10 recommendations that RISD put in this critical review. So we are working in the scale up of the technologies. We are working on include tests on real wastewater. We always did it with real wastewater. We are investigating the effects on bacteria, microorganisms in general with whole genome sequence and 16x sequence and counting plate as well. We are investigating the effect on antibiotic resistance determinants, taking the possibility like the transfer to mechanisms with the plasmids and as well. And we are investigating the efficiency process and the impact of the treatment as well with the toxicity response. Nowadays, we have collaboration with Maria Clara. She is here with us, and we can investigate it with different kind of organisms. If our treatment is not only effective to remove contaminants of emergent concern, but also didn't lead or didn't cause toxicity for the environment. So I would like to say thank you for everyone. Obrigada. I would like to say thank you for my research group. I have amazing people, amazing researchers working with us. We are proud to say that we are a Brazilian team. We have some small collaborations abroad, but we are almost 100% Brazilian team. And we are doing all of the best to like put this the subjects like real concern, this challenge for antibiotic resistance within our country. It's important for us because it's far from our reality to talking about genes and bacteria in wastewater because we have other like common and not it's less important problem, but it's like common problem to treat wastewater. But I think that we are starting some things that could stay there for like prevent the spread of genes and antibiotic resistance genes and bacteria in our country and around the world, of course. So I would like to thank the funding of this project, the Bill and Melinda Gates Foundation, the CNPQ agents in our country, the health ministry in our country as well, my institution, and I am able to have the questions if you have.
So Camila, thank you for this wonderful presentation. It was a really great presentation, giving an overview of the topic and also results you've been getting in your research group. We do have a first question from Despo. She thanks you for your great talk and the remarks for her work and some of her colleagues. She congratulates you on the important work and publications you're doing. And she has one question. So have you performed the experiments using also lower concentrations than 30 milligrams per liter of iron? Why did you use so high concentrations of iron? And what is the limit for iron disposal in treated wastewater in Brazil? She had to leave, but her student is still here, so he will tell her the answer afterwards.
Okay. Thank you for this question. It's interesting questions as well. And now I think that some of referees did the same. We used this strategy to allow that the concentrations of iron still remain in the environment or in the median, in the reaction median. So we use 30 milligrams. We divide these concentrations in like intermittent iron additions, like 15 in the beginning and 5 milligrams after maybe 30 minutes, and then more 5 milligrams, and the total is 30 milligrams of iron. And then in our country, we don't have problems with the disposal of this kind of effluents because we have limits only for dissolved iron and not for total iron. So total iron concentration. So for dissolved iron concentration, we have less than 5 milligrams per liter, and this is our limit iron for disposed treated wastewater in Brazil. But of course, if we could investigate it for reuse, like in the crop and agricultural areas, we have to investigate it if this total iron concentration could damage the crops or could interfere in the soil, in the like salinity, or maybe in the soil quantity of iron. For us, it's not too much because we are in the mining state in our country. So our country is well known about the very mining companies that we have because we have naturally high concentrations of iron in our soils. So we believe that this could be not interfere a lot because we have very high concentrations of these metals in our soils, nature in our soils as well. So thank you, Despo. I know that you are not here anymore, but thank you to be so kind. And for us, it's really good to have you and your research group here because we really like the uh, the papers and the works that Despo and with her research group publish.
Yes, it's in our, but on the side of our beds, we read them before going to
Yeah.
sleep, almost. So we don't have any other questions here yet. I did remind participants that we still have a few more minutes. I don't know. Cesar Pulgarin just raised his hand. So Cesar, welcome and thank you for being here. You're welcome to ask your question. Maybe you might want to open your microphone.
Beautiful presentation, very didactic presentation. Congratulations. I have a little question.
Thank you.
How do you, what is your explanation about the differentiated response of the genes taking in account the fact that the hydroxyl radicals is normally able to attack all the organic matter except in 2 or 3 molecules, how could you explain this differentiated response?
The differentiate response according to the genes you are
The genes,
asking.
the genes,
Yeah,
yes.
according to the genes. Yeah, you are talking about that. We have the hydroxyl radicals that can attack like other constituents of the matrix, like organic matter, because we have organic matter as well. But we believe, as you investigated, that we have other mechanisms involved, and not only for radical oxidative species like attacking the bacteria and achieve the genes. I believe that we have other intracellular mechanisms that are causing this eliminated, but how what is the kind of genes is more susceptible or less susceptible for photophanol attacks or for hydroxyl radical attacks? We didn't identify. We need the help for microbiologists. Do you know what I mean? Because I'm not biologist, and I have some collaborations with Maria Clara. It's a biologist, and other people in the group. But we need to investify properly this point. You are right.
Thank you very much. Congratulations again. Thank you.
Thank
Thank
you
you,
so much.
Professor. Thank you, Professor Pugari, for your question. We actually have another question here, Camila, from Paula T. Rivera. I don't know if I've pronounced it correctly. There's someone with the microphone open. I would ask you to close your
It's an
microphone, please.
angel.
Okay. So
Okay.
Paola Tirira is asking you, she congratulates you, and she asks if there are any investigations with photoelectrophantom, solar photoelectrophantom.
Oh, sorry. Just didn't hear the last part. Are any investigation with photoelectrophantom solar? No, Paula. Unfortunately, we never investigated the the photoelectrophantom solar electrophantom. It will be a very good option to investigate it as well, but we never did it. If you have some results to share with us, will be great to learn more about this process and how this could um, like could combat or could deactivate genes and remove bacteria as well.
Yeah, and then we have another one from Angel Joseph. How do you quantify hydroxyl radicals?
We didn't quantify reductive radicals. We didn't. I know that there is methods for that, but we just quantify the consumption of hydrogen peroxide, but just to make sure that we have enough hydrogen peroxide and we are decreasing the quantity because we don't want to discharge the effluents with high levels of hydrogen peroxide, but in fact we didn't quantify the hydroxyl radical inside of the matrix. When you inside of the medium, when you are working with real matrices, it's kind of difficult to have like this analysis because we have so many interference in the matrix, like in real matrix, wastewater matrix. There's only secondary effluents. We have less quantity of soils, but we have other constituents, and we didn't quantify that. Bulgari again?
No, I think he forgot to.
Okay.
Yeah, his hand raised. Well, we don't have any questions. Luigi Rizu is thanking you for your really interesting presentation. Your work on the effect of photo-driven AOPs on antimicrobial resistance is significantly advancing the knowledge in the field. Congratulations to you
Luigi.
and your collaborators. Do you want to
Thank you,
comment?
Luigi. And thank you, Luigi, to allow for us to investigate it and to discover more, because you contribute a lot in this matter of antibiotic resistance bacteria and genes. And I am just like a junior, and you are like a monster with Spulgarin and Despo in these subjects. And I'm really happy that you stay here with me in this presentation.
Yes, and it's really nice to have compliments coming from him. You might be very
Yeah,
happy right now.
yeah.
So Angel Joseph is asking another question. What about the toxicity of intermediate compounds formed, if
Yes.
any?
Yes, and this work that have published, we didn't include the toxicity analysis because at this time we didn't have more than microtoxicity, like microcystis and microtox analysis. But nowadays we are doing the DAFNA microtoxicity with microcystin. Maria Clara could help me if I am wrong, and half the cells and Yalela as well. So we are working in algae, uh, bacteria and other organisms to identify the toxicity. In our last results, it was not present and it was published yet. We identify that the most part of the solar treatment could remove the toxicity. Some parts of when you use different kind of oxidants, this could we can have better removal of toxicity, but we didn't publish this result yet, but we are investigating as well. And the last results from a master student that she worked in the hospital wastewater, and she applied the ozonization, and she got good results to removal bacterias, and we didn't identify genes, but resistance bacteria and toxicity as well. And about the intermediate compounds formed, we are not working on this direction with real matrices. In fact, to be honest, we are working in this direction, in this matter, because we're working not only to investigate the removal of bacteria and resistance genes, but we are working on the removal of antibiotics as well. So when you are investigating if the antibiotics are being removed, and even if the bacteria and genes are being removed. So I'm to investigate it if we are forming byproducts, but it's not published yet. It's difficult because it's a very long way for analytical methods when you are working with the real matrices. So
Yes, it is. It's challenging working with
it's a
real
challenge.
matrices, but it was our decision because we think it's closer to the reality and maybe we can get closer to application, especially in our country where we
Yeah.
only go up to disinfect to secondary treatment. So it might end up if we have the chance to apply this after secondary treatment. So we lose some points, like in identifying intermediate compounds by working with real wastewater, but we get closer to application in real scale. So, well,
Yes.
I don't think we have more questions. So I'm really glad to have had the chance to be the moderator of this webinar today. I thank Professor Camila for her talk and for all the opportunities she's been giving to our students and to the department and the projects she's been leading. And I thank all the participants for being present here today, for all the interesting questions. And I hope to see you in the next webinar. Bye-bye.
Okay. Bye-bye.
Oh,
Thank
you're
you.
going to receive certificate for your participation, okay?
Yeah, just to download in the list of participants, Maria Clara,
Yes,
I
I
think,
did.
and send. Yeah.
I
Okay.
did.
Maria Clara, I would like
Yes.
to kindly ask you to send me the link to the video, okay?
Okay.
When you upload,
Yes.
so we can upload in the school
Correct.
YouTube channel, okay?